Hello everyone, welcome to another session on the unit Diversity in Living World. Here we are going to discuss about the next division of the plant kingdom, that is Bryophyta. So let us first look into the key concepts of this session. Let us see what are the contents of this session. Here we are going to discuss about the general characteristics of the division Bryophyta and the two important classes of Bryophyta, that is Hepaticopsida and Bryopsida. Then we will discuss about the importance of Bryophytes. In the beginning, we will see what are the important characteristics of the plants which belong to the division Bryophyta, which are commonly called as Bryophytes, which are rightly regarded as the amphibians of plant kingdom. So these are called amphibians of the plant kingdom because these are the first terrestrial plants. These are the first terrestrial plants that are evolved, but still, even though they are terrestrial, they require water. They require water during fertilization. Hence, they are called amphibians of the plant kingdom. This is a very small group of plants which includes 850 genera and 24,000 species of plants. All the plants which belong to bryophytes, they are again non-vascular, just like algae. Even these are also non-vascular. That means non-vascular means they lack the vascular tissues, the conducting tissues, that is xylem and phloem. They are also non-flowering. They do not bear flowers. They are non-seeded. They don't even produce seeds. So we can call them as cryptogams. But they are embryophytic plants. These are the first terrestrial plants to evolve on Earth. And if they are terrestrial, they actually need shady places for their growth. Hence, they are also called as sciophytes. That means the plants which grow in shady area. Hedwig is rightly regarded as the father of bryology. Bryology is study of bryophytes. Then Shivram Kashyap is rightly regarded as the father of Indian bryology. In bryophytes, the dominant phase in the life cycle is actually the gametophytic life cycle, and this gametophyte is independent. The gametophytic plant body of the bryophyte is independent. So what is actually gametophytic plant body? The plant body which can produce the gamete, from which the gametes are produced, it is called as gametophytic plant body. And gametophytic plant body is always haploid. So the haploid gametophytic plant body is green, photosynthetic and independent in case of bryophytes. But still, the plant body is not completely differentiated into root, stem and leaf. Still, the plant body is a primitive type where you cannot find the actual roots, stem and leaves, but instead root-like structures are present. These root-like structures are called as rhizoids. The rhizoids may be either unicellular or they may be multicellular. Unicellular rhizoids are present in Markantia and Rixia. This is very important for competitive. Unicellular rhizoids are found in Markantia and Rixia, and multicellular root-like structures are present in mosses. And what is the function of rhizoids? Rhizoids help in the absorption, fixation and absorption. Okay. Then the plant body is still thalloid. Thalloid means flattened undifferentiated plant body which is present in algae. The same thalloid plant body, still it is present in the lower bryophytes, which are called as liverworts. The lower bryophytes are still thalloid, whereas in higher bryophytes they have a leafy structure. In higher bryophytes, the leafy plant body, that is in mosses, the leafy plant body is differentiated into multicellular rhizoids for absorption and an erect shoot. This erect shoot, it is actually made up of spirally coiled leaves, and the leaves are actually called as the microphylls because the leaves are very tiny and they are spirally arranged to form an erect leafy shoot in case of mosses. These bryophytes during rainy season, they completely cover the soil surface. They look like mat on the soil. They undergo vegetative reproduction by fragmentation of the thallus, and each fragment can give rise to a new plant body, or the vegetative reproduction through gamma cups. So here in the picture you can observe the gamma cups which are present on the dorsal surface of the liverworts, that is the upper surface of the liverwort, such as Markantia, and each gamma cup has various gemmae. So these gemmae are actually in the shape of 8. These gemmae are in the shape of 8. There are 2 to 8 gemmae in each gamma cup. And vegetative reproduction is through tuber formation in case of Anthoceras, which is another type of lower thalloid bryophyte. Then sexual reproduction involves the formation of gametes, and the gametes are formed in specialized sex organs which are called as antheridium and archegonium. And in case of bryophytes, the sex organs are jacketed. You can see the covering on the cells which produce the gametes. They have jacketed sex organs. The male sex organ is called antheridium and the female is called archegonium. Both are antheridium and archegonium. If they are present on the same plant body, then such type of bryophyte, they are said to be homothallic. Homothallic when both male and female sex organs are developed on the same plant body. Or the bryophytes can also be heterothallic, that is when male and the female sex organs are developed on individual plants of the same species. Individual plants. This is formed on one individual plant and the female sex organ is present on a different plant, but both of them belong to the same species. Then such type of bryophytes are called heterothallic. Example is Markantia. The fertilization in bryophytes, it is said to be zooidiogamy, zooidiogamous type of fertilization. Zoo because the male gametes are motile. They have 2 apical flagella. They are motile and they swim in the water during fertilization. Hence it is called zooidiogamy. After fertilization, the zygote develops into embryo. I told you these are the first plants to bear the embryo, therefore they are the first embryophytes. The embryo later develops into a sporophyte. The sporophytic plant body, it is differentiated into the sporophyte is differentiated into foot, seta and capsule. So I'll show you a picture how the sporophyte look like. So this is actually the sporophyte of the mosses. This is a gametophyte. You can see the gametophyte forms a mat on the surface of the soil. On the gametophyte, the sporophytes are dependent, and the sporophyte involves a seta and the basal part with which the seta is attached to the gametophyte. This is called as food, and a capsule Okay. So the sporophyte is differentiated into food, seta and capsule. The sporophyte is completely or partially dependent on the gametophyte. This is very important sentence. The sporophyte is dependent on the gametophyte in bryophyte. The sporophyte is completely dependent in case of liverworts, that is Mercantia. The sporophyte is completely dependent both for food and shelter. Whereas in mosses, the sporophyte is green in color, so it can photosynthesize, but for shelter it is dependent on the gametophyte. So the sporophyte is completely or partially dependent in bryophyte on the gametophyte. In the sporophyte, there are spore mother cells. Inside the sporophyte, that small structure that we have seen, there are spore mother cells. These spore mother cells present in the capsule of the sporophyte, they produce spores upon meiosis, which is actually haploid. So after fertilization, the zygote develops into embryo, and zygote is actually 2n. Okay, so the embryo is also diploid, that is 2n. The sporophyte is also 2n. The sporophyte contains capsule. The capsule has spore mother cells which are also 2n. These spore mother cells will undergo meiosis inside the capsule and give rise to the spores which are haploid. Okay, and these spores upon germination again give rise to gametophytic plant body directly. The gametophyte is formed in liverworts, but in mosses it involves intermediate stages. First, the spore develops into a juvenile stage. This juvenile stage is called as the protonema. I'll show you in the picture. And later the protonema develops into a leafy stage of the moss. The life cycle in bryophyte is haplodiplontic because the predominant phase is, which is a predominant phase in bryophytes? The predominant phase is the haplontic gametophytic phase, and upon the gametophyte there is a sporophyte which is dependent. The predominant gametophyte alternates with the sporophytic generation, hence it is called as haplodiplontic life cycle. Okay, so the main independent plant body is gametophytic, so we call it as haplodiplontic. Otherwise in pteridophytes we call it as diplohaplontic. Here in the picture you are able to see the development of the antheridium upon which the antheridium are present. So here inside this will be the antheridium. The antheridium develops on a stalk which is called as antheridium. The antheridium is developed on one plant, on one thallus, and Archegonium on the other thallus. Therefore, we can say that Mercantia is heterothallic. So the female reproductive structure, that is Archegonium, is developed inside this structure, okay, which is present on individual plants. So these are actually the gamma cups through which vegetative reproduction occurs in Mercantia, and you can see they are present on the dorsal side. In Mercantia, The plant body is thalloid, flattened plant body, and on the dorsal surface you can find thick ridges which are called as the midribs, which have parenchymatous tissue for conduction. Okay, and you can also observe the dichotomous branching. So every branch is once again divided into two parts. You can observe dichotomous branching in liverworts. So these are characteristics of liverworts: flattened plant body, then dichotomous branching, presence of a thick midrib on the dorsal side, and on ventral side you can also observe unicellular rhizoids. And the gamma cups are present near the notches on the dorsal surface. Marchantia are heterothallic with Anthridium and Archegonium developed on different gametophyte plant body of the same species. This is the structure of Anthridium inside that Anthridiophore upon which the Anthridium is present. Each Anthridium is jacketed. You can see the jacket of Anthridium. It encloses a number of androgonial cells, and such 2 to 4 Anthridium are present in one crevices upon that Anthridiophore. They are surrounded by Paraphysis, that is the sterile hyphae, and leaves which are called as perigonial leaves. The Anthridium are surrounded by perigonial leaves and paraphysis, which are sterile hair-like structures. So these perigonial leaves, they are directed in the outward direction. That helps in the easy dispersal of the male gametes. In each Anthridium, there are hundreds of androgonial cells. So these are individual androgonial cells. Each cell in the androgonium, it has to undergo cell division. You know, it is the gametophytic plant body, so the androgonial cells are haploid. This haploid androgonial cells, upon mitosis, they produce antherozoids. They produce antherozoids upon mitosis, and these antherozoids, they are motile and they swim towards the archegonium. Here you can see the structure of antherozoid. It is spirally coiled, okay, with two long apical flagella. So these antherozoids are produced by the androgonial. So one Anthridium produces thousands of antherozoids inside the structure of the Anthridium. Then in the Archegonium structure, each Archegonium contains a stalk, a swollen base which is called as the venter, and an elongated structure called neck. The neck is very long and it contains neck canal cells. So these are neck canal cells, you can see. So these neck canal cells, they are 4 to 10 in number. They are 4 to 10 in number. And these neck canal cells, they produce chemical substances which actually attract the antherozoids towards the archegonium. In the swollen venter, there is one venter canal cell and one large cell which is actually the egg or the female gamete. Okay. So during fertilization, the antherozoids, they come through the neck by disrupting the neck canal cells. After the disruption, they enter into the venter where they fertilize with the egg. And archegonium are surrounded by pericaceal leaves and once again sterile hair-like structures which are called as paraphysis. So in this picture, we can understand how the fertilization and development occurs in the first division of bryophytes, that is Hepaticopsida, which includes liverworts. Example for liverworts are Marchantia and Rixia. They are called liverworts because the thallus of these plants, they resemble the liver lobes, and hence they are called as liverworts. So in Marchantia, we know that the male and the female gametophyte are different. They are heterothallic. Okay. Vegetative reproduction occurs through gamecops. They have midrib. Unicellular rhizoids are present in the Hepaticopsida or in the liverworts. During sexual reproduction, the male gametophytic plant body, it has a archegonial head. Inside the archegonium, on the lower ventral surface, they produce a number of archegonium. So they are actually suspended in the downward direction. And the male plant, which is having the antheridial head, this antheridial head bears a number of antheridium. Antheridia is plural. It has a number of antheridium. In the antheridium, there is formation of motile antherozoids. You can see these antherozoids, they swim towards the archegonium, and fertilization occurs in the venter. The egg fertilizes to develop into zygote. Zygote gives rise to embryo, and the embryo develops into a sporophyte. But the sporophyte in liverworts, it is highly reduced. Liverworts have highly reduced, means very small sporophyte is present in liverworts. Okay, and in the sporophytic structure, they have a number of spore mother cells. These are spore mother cells which develop into spores. Along with this, they have other cells also, which are called as elaters. Elaters are the cells which are present in the sporophyte of the liverworts, which help in the spore dispersal. So what we have to understand here is gametophyte is haploid. Archegonium and antheridium, they are haploid. Egg is haploid. Antherozoid or the sperm is haploid. But after fertilization, the egg becomes zygote, which is diploid. That develops into diploid embryo. The sporophyte are diploid, and all the cells inside the sporophyte, including the elaters and the spore mother cells, are diploid. And now in the spore mother cells, the meiosis occurs that helps in the formation of spores. These spores are haploid. After meiosis, the spores which are produced are haploid. Upon germination of the spore directly gives rise to the gametophytic plant body in case of Hepaticopsida or liverworts. Okay, then in case of another division of Bryophyta, that is Bryopsida, which includes mosses, such as Funaria, Sphagnum, Polytrichum, etc. In case of mosses, you can see here, these are gametophytic plant body. Okay, so the gametophytic plant body is haploid, and you can see the development of both male and female reproductive structures on the same plant. So I told you mosses are generally, they are homothallic. Then in antheridial head, there are various antheridium which produces the antherozoids, and in the archegonium, they have in the archegonial head, they have number of archegonium with neck and venter, where the venter contains actually the egg. Upon fertilization of the egg by the sperm, there is development of zygote. The zygote develops into embryo. Okay, the embryo gives rise to the sporophytic plant body. You can see here the development of the sporophyte from the embryo. The sporophyte differentiates into foot, seta and capsule. Okay, and during the development, the venter, the neck completely gets degenerated, and the venter, it becomes thin, membranous, transparent membranous structure which is called as calyptra. Upon maturation of the sporophyte, the calyptra will fall down. So calyptra is a part of actually the gametophytic structure. Therefore, calyptra is actually haploid. These are competitive questions. We think that calyptra is on the sporophyte, so it is a diploid in nature. But actually calyptra is derived from the venter during the development of the embryo. So this venter covers the developing sporophyte for protection. Hence calyptra, it is actually haploid in nature. Then the developing sporophyte, you can see here, has foot, seta and capsule. Inside the capsule, there are spore mother cells which give rise to the spores. Upon germination of the spores, you can see the germination of the spore here, there is development of initial filamentous structure. The filamentous structure, the juvenile structure or the juvenile stage or the filamentous stage of mosses, it is called as the protonema. It is called as protonema. In this juvenile structure, there is development of bud. These buds are called as secondary protonema. These buds develop into young leafy gametophytic structure. Again, gametophyte gives rise to the male and female Archegonium and Antheridium respectively, and the life cycle continues. So here actually the sporophyte is green in color and can perform photosynthesis but still remain attached to the gametophyte. Hence it is partially dependent on the gametophytic plant body. In this picture we can observe how in external view the sporophyte looks like on the gametophyte. They have a foot which is embedded in the gametophyte, a long stalk which is called as the seta, and this is actually the capsule. You can also see the calyptra which is covering the capsule here. Okay, but here the calyptra is not there, it is already fallen off. The calyptra falls down after the maturation of the sporophyte. This is lateral section of capsule in Funaria. In Funaria, you can observe in the sporophytic structure, there are a number of peristomial teeth. These are hygroscopic and help in the spore dispersal. In liverworts, it is elaters that help in spore dispersal. In case of mosses, it is peristomial teeth. Due to the hygroscopic nature and the movement of the peristomial teeth, there will be dispersal of the spores. This is in turn covered by a door which is called as operculum. Okay, and inside the sporophyte, there is columella tissue for nutrition, which in turn it surrounds the spore sac. These spore sac contain, inside the spore sac there is spore mother cell. These spore mother cells, they have undergone meiosis to develop into haploid spores. There are also air cavities for exchange of gases, and the strands in the air cavity are called trabeculae and the air cavities are called lacunae. and the swollen part of the tip of the seta, that is the end of seta, and the basal portion of the capsule which is swollen is actually called as apophysis. So here apophysis or seta or the spore mother cells, that is sporogenous tissue, columella, peristomial teeth, operculum, all these are diploid. Okay, but the spores which are produced from the spore mother cell after meiosis are haploid. You can see the operculum, the door of the capsule, and this will be in turn surrounded by the calyptra, which is actually the part of the venter. After the maturation of sporophyte, first the calyptra falls down, then the operculum also falls off, then the peristomial teeth, they are going to contract, and there will be dispersal of the spores from the peristomial teeth. So this is how the spore dispersal occur in the Bryopsida. But very important in case of Bryopsida is you have to remember the stages of development in the spore, the juvenile phase, which is called as protonema, and it is filamentous in nature, which gives rise to secondary protonema, that is the bud which develops into a leafy bud which develops into young gametophytic plant body. Okay. Now coming to the importance of bryophytes. These bryophytes, since they cover the soil like mat, they hold the soil particles tightly, therefore prevent soil erosion. The rhizoids, they hold the soil particles and prevent the soil erosion. They also help in soil formation in the ecological succession. After lichens, the next developed plants in ecological succession are actually bryophytes. Therefore, they also help in withering of rocks and soil formation. Then a bryophyte called sphagnum, it has maximum water retention capacity, 18 to 26 times more water it can retain. Therefore, it is used in packaging of flowers, and it can be used as a substitute to the cotton. Hence, it is also called as cotton moss. At the same time, sphagnum is also useful in the formation of peat, which is a type of a fuel, fossil fuel, and this peat is used to run the engines, and hence sphagnum, it is also called as peat moss. It is also called as peat moss. Another important useful aspect of bryophytes is polytrichum. It is used in the treatment of kidney and gallbladder stones. The decoction of polytrichum is consumed to cure kidney and gallbladder stones. So these are the economic importance of economic and ecological importance of bryophytes. In the next class, we are going to discuss about the rest of the divisions of plant kingdom, that is pteridophytes, gymnosperms and angiosperms. Thank you.